Okay, so we just finished today's topic about expression vector. Okay, so later on, if you have time, then you should read a book that the corresponding pages that I list on the syllabus about the topic. All right, and now I'm going to switch the topic to the some experimental procedure. And yeah, the class material, that's the yes, that's I posted on the Blackboard that has everything you need. Okay, and so now we will discuss the some experiment. All right, so this experiment supposedly that you will perform in the laboratory, but because during this period of time we can only do it online, so we will go through this step by step slowly. But for some experimental procedure in the future, we will show the video and then to go through the video to tell you the detail. And also you will remember need to perform the lab simulation. Okay.
Oh, Professor, for the lab simulation, how am I gonna, like, what are we supposed to do for the lab simulation?
Sorry, what did you say? Can you say louder? I cannot hear you.
Oh, I'm sorry. For the lab simulation, what am I supposed to do exactly? Like, what are we supposed to do in the lab simulation?
What are you supposed to do? You were not here in the first 10 minutes of the class, right?
Yeah, I wasn't here. I was trying to, like, because I couldn't see from where would I, like, log into the video. I was trying
Okay,
to
so for the labs, okay, since we are recording, let me finish the whole class, and then we will go back to that again, all right?
Okay, thank you so much. Thank you.
Okay,
Sorry about that.
good. So for the rest of the week, okay, I will do the demonstration how to design, how to use the DNS software, and how to design the gene knockout project. All right, and this will be useful for you later on. That each of you will have a specific project to perform the gene cloning, and so you will need to learn how to do this. Okay, so our project, we will use yeast as our model organism. So the reason we use yeast is because yeast is easy to grow, and the yeast has a small genome size, only 12 megabase, and 16 linear chromosome. And but although the yeast is the simplest eukaryotic cell, but it displays most of the feature of the higher eukaryotes. So it's and also there are many powerful genetic and molecular tools available for the yeast. So that's why a lot of experiments are performed in yeast so that we can understand the eukaryotic cell. And for the yeast, the open reading frame is about 6,500, and the verified open reading frame is about 5,100. Okay, and then for the knockout project, okay, the cloning vector, we are going to use the PRS-413, right? PRS-413 is a centromere plasmid, okay? And the PRS-413 is the centromere vector with a history marker. History marker, that means this vector carry the gene that can produce the histidine, and then we have to use the cell. We have to use the cell. It's autotrophic mutant. The history autotrophic mutant. That means the host that we're going to use would be the cell that carry the history mutant. That means they cannot produce the histidine. All right, and so we use this wire, the PRS413, all right, as the vector. And then for this example, okay, I'm going to do the demonstration to knock out the HSK2 gene from the chromosome. Okay, the HSK2 to knock out the we are going to knock out the HSK2, all right? And then before we continue, all right, so sometimes you need to do some literature search. So for example, you want to understand the HSK2, what is this gene? Then you have to go to the literature search, all right? So this is literature search. The example you use the PubMed, all right? Use the PubMed to do the perform the literature search. Perform a literature search so that you'll be able to understand the gene that you want to knock out, right? Because when you knock out a specific gene, then you can compare the wild type and the mutant so that you will know the function of that specific gene, okay? Because after you knock out that specific gene, then the cell will behave differently from the wild type. And so that you understand the specific function of that specific gene. And so now I'm going to do some demonstration, right? And then I'll be no, I will assign a specific project for each of you later, okay? But for this week, I will do the demonstration, all right? But step by step, so that you have to learn. And also tomorrow, I will tell you a specific DNA software that you need to download so that you will be able to perform the DNA manipulation, okay? So now, let me stop this. Stop this. So, just a second. So I just click the link, right? That brings us to the PRS413, all right? PRS413, and then this is the plasmid map of the PRS413. And then you can see here, you see this one, the centromere sequence and the autonomous replicating sequence here, all right? The CEN-ARS, right? Centromere fused to an autonomously replicating sequence, right? So that you can duplicate the plasmid, right? And then we have the HIS3 promoter and the HIS3 gene here. Did you all see this plasmid now? Okay, so for the PRS413, all right, I'm going through this feature of this plasmid, okay? So for the plasmid DNA, in order for the easy understand the plasmid, normally you will see in the circular DNA here, you have a symbol here, and this will be labeled as number 1, okay? On the top here, that will be number 1, and then you go from the clockwise direction, right? And then so if 1 and all the way to the end, so the size of 4970. So the last sequence here will be 4969, all right? And then 4970, okay, 4970. And then the first one you will see is the HIS promoter, right? And the HIS gene. And the HIS gene here is between 504 and the 1163. And the size is 660 base pair long. and 219 amino acid. All right, that's 504 to 1163. And then the VECT, the promoter is 317 to 503. 317 to 503, all right, and then 504, that's an open reading frame, all right, and then start this history that this plasmid produce the histidine. So that can help you, this is a selectable marker, help you to select the cell, all right? And also, in this plasmid, we have this LEGZ gene, that's 1661 to 2239, all right? That's 579 base pair long, LEGZ gene. And inside the LEGZ gene, we have this multiple cloning site. See, the multiple cloning site MCS is 2076 to 2183, which is inside this LEGZ gene. So that means if you clone something inside here, that will inactivate the LEGZ expression, okay? And then we have this original replication site, okay? And then you can see it's a high copy number, and this original replication. This is for the bacteria. And the ampicillin resistant gene, all right? So the location of the ampicillin resistant gene is between 3396 and the 4256. Okay, and then this is 4398 to 4901. That's a centromere sequence and the autonomous replicating sequence. So this is the plasmid map of the PRS413. Okay, and this is a shuttle vector because it can be replicated in the yeast cell and also replicate in the bacteria cell. And here is the here is the sequence. Now they show you the sequence, all right? And of course, after tomorrow, after you download the sequence, then I will also demonstrate how to use the DNA software to do this, to manipulate the sequence. So this is the full sequence of the PRS413. As you can see, this is the history promoter, all right? And this is a history open reading frame. And all these are the restriction enzyme cutting site, okay? And then with original replication and LEGZ gene and multiple cloning site here. Here's the original replication. And then we have these ampicillin resistant gene. And then centromere and the autonomous replicating sequence, right? So this is the general feature of the PRS413, all right? Just give you some idea that what we are using, all right? And then we will do this, I think it's Wednesday, okay? Tomorrow I will show you how to download the software, and then Wednesday we will start using the software. And then the PubMed. Okay, so this is the website that you need to use for all the literature research. So for example, if we want to study the HSK2, right? So we can type the HSK2, all right, and perform the search. So that give you some information about the HSK2. And of course, if you want to get the full text, right, you can get the free full text. And then if you want to get the review, so that you can have more specific, you can get the detail. So you get the review. So that now, for example, the first one here, the FAMS microbiology review, the early step of glucose signaling in yeast. Okay, you can click these, so that can tell you, all right, so this glucose phosphorylating enzyme HK2. And then you can go to the full text, and then to study you can download a PDF, and then to read about the HSK2. And then another source would be another source, because the PubMed will give you more detailed research information about the specific information you want to get. And then another easy way is to go to eastgenome.org. They summarize the detail for you already, all right? If you type HSK2, then you hit enter, then they give you this overview of the specific gene, HSK2, hexokinase isoenzyme 2, phosphorylate glucose in cytosol. All right, phosphorylate is the glucose in cytosol, so that the glucose will be able to transport into the cell. That's the specific function of HSK2. All right. And predominant hexokinase during the growth on glucose repress expression of HSK1, right? So this eastgenome.org will give you the overview of the sequence, the gene, and also you'll be able to download the sequence, which I will also demonstrate later. Okay, this is just to show you, and it's in chromosome number 7. You see the chromosome number 7, and location is 23,935, 25,395. That's the location of the hexokinase isoenzyme to HSK2. The length is 1461. Okay, that's the location, and then you can download the sequence. All right, that I will show you later on. Okay, not today. So basically, and all the detailed information of the HSK2, then you can go through these. And this is summary paragraph. It's more detail about the HSK2, and also they give you some references. Okay, and to reference about this gene, then you can, if you are interested in this, you can even go to go to these references to see the detail. All right, so that's the HSK2. So are we all good for now, until now?
Yeah.
That we will